In order for life to continue, genetic information must be transferred from one generation to next, and cell division is the process that facilitates this transfer of genetic information. And these processes include things like binary fission, mitosis, and meiosis. Now, meiosis is a process that produces sex cells, such as sperm and egg cells, and these are also referred to as gametes. Mitosis, on the other hand, produces body cells. Otherwise known as somatic cells. So all the other cells in your body that aren't sex cells. And this is for things such as growth, repair, etc. Now these two processes are related in humans. So for example, in humans, we produce sex cells, or gametes, through meiosis. And these cells are haploid, meaning they only have one set of chromosomes in each of the egg and the sperm. So N equals 23 in the case of humans. Now, when a sperm fertilizes an egg, a cell is produced that has a full set of chromosomes, which is referred to as a zygote. So a fertilized egg is a zygote, and this fertilized egg is, of course, diploid, because it has one set of chromosomes from the sperm and one set of chromosomes from the egg. So we'll have 46 chromosomes in total. Mitosis then results in cell divisions so that we can grow into mature adults and then produce gametes through meiosis, and the process continues again through a cycle. The sequence of events from one cell division to another is called the cell cycle. The stage in which cells are actively dividing and producing new cells is called mitosis, represented by this red portion of the cycle here. And as you can see, it's only really a small portion of the cell cycle. The remainder of the cycle, represented in blue here, is called interphase, and that's when the cell is either doing its job, its intended purpose as a brain cell or a skin cell, and or preparing for mitosis. Both interphase and mitosis can then be broken down into smaller phases where the cell is doing different things. So interphase, for example, the first stage of interphase is called G1. This is where the cell is actively growing, doing its job, as well as preparing for DNA replication. The second stage of interphase is S phase, where the DNA is actually replicating, getting ready for cell division. In G2 phase, the cell prepares for cell division, and that's the final stage of interphase before mitosis occurs. The very final stage of cell division after mitosis is called cytokinesis, and that's where one cell actively splits into two cells following mitosis. In addition to these 5 phases, there is another phase called the GO phase. And the GO phase indicates a stage in which cells are not actively taking a part in the cell cycle. And this occurs with cells that are really highly specialized, such as nerve cells, which are described as being in GO phase. They're just doing their job. They've withdrawn from the cell cycle. However, some cells in GO can actually re-enter the cell cycle under certain circumstances. DNA replication occurs during the S phase of the cell cycle, and the DNA in the original cell is called the parental DNA, and the parental DNA needs to duplicate to create 2 copies of the DNA for cell division so that each new cell has a full set of DNA. The parental DNA is split apart by an enzyme called helicase. And helicase breaks apart the bonds between the nitrogenous bases of the double strand, resulting in 2 single strands of DNA. The junction between the unwound single strand and the double helix is called the replication fork. The replication fork moves along the parental strand, unwinding the DNA strands. There are free nucleotides in the nucleus that then attach themselves to the 2 strands of unwound DNA with the help of the enzyme DNA polymerase. Another enzyme called DNA ligase then seals the new short stretches of nucleotides into a continuous strand that then rewinds. The outcome is 2 double helix DNA strands that consist of 1 parental strand from the original cell and 1 new strand. In this way, one of the 2 strands of DNA is a parental strand which is retained from one generation to the next, while the other is a replicated strand that is new. Because of this process is called the semi-conservative model, where one strand is newly formed and the other one is an original strand. Both the volume of a cell and the amount of DNA in a cell changes during the cell cycle. This first graph shows how the volume of a cell changes over a time during the cell cycle, and it refers to a mother cell and a daughter cell. What that means is that the original cell before cell division is the mother cell, and the cells that it divides into are referred to as the daughter cells. This period of time here represents one full cell cycle, from interphase through mitosis and then finally cytokinesis, where the mother cell divides into 2 daughter cells. And as you can see, there is a steady increase in the cell volume over this period of time through interphase and mitosis. And then finally, as cytokinesis occurs and cells divide, there is a decrease in the cell volume. And this makes sense. The original cell, the mother cell, has to increase in volume as it grows, but also makes room for duplicated DNA. At this point here in time, cell division occurs and the volume of the cell then decreases so that each daughter cell that results from this cell division has the original volume of the mother cell. And then the process repeats itself again with each daughter cell. This second graph here shows how the amount of DNA changes throughout the cell cycle. What you'll notice is that during the G1 phase of interphase, there's absolutely no change in the amount of DNA in the cell. And that's because the cell has one of each chromosome that is unduplicated in that cell. It's only during the S phase that there is an increase in DNA. That's because DNA is duplicated during the S phase, getting ready for cell division. That means that during the G2 phase, there is actually 2 copies of each chromosome. So this is still one chromosome here, but it is duplicated. A duplicated chromosome is referred to as having chromatids. So each one of these is a chromatid, and they're joined together by a centromere. These chromatids are referred to as sister chromatids. Now, when the cell divides, these sister chromatids are pulled apart into the 2 different daughter cells, and that is shown here with a decrease in the amount of DNA during cytokinesis, when the mother cell, which had 2 copies of each chromosome, has now divided so that each daughter cell then has one copy of each chromosome.